Bioinfocongress II Profesör Doktor Duygu Uçar İnsan Bağışıklık Sistemi'nin genomik imzaları, yaşlanma ve aşı yanıtları. Profesör Doktor Duygu Uçar, lisans eğitimini İlkent Üniversitesi Bilgisayar Mühendisliği bölümünde, doktor eğitimini Ohio State Üniversitesi'nde aynı alanda tamamlamıştır. Doktor eğitimi süresince Danimarka Teknik Üniversitesi Biyolojik Dizi Analizi Merkezi'nde misafir araştırmacı olarak görev almıştır. Doktor sonrası çalışmalarını Iowa Üniversitesi'nde Ulusal Bilim Vakfı Bilişim İnovasyon Bursu tarafından finanse edilmektedir ve Stanford Üniversitesi Genetik bölümünde tamamlamıştır. 2003 yılından beri Jackson Genomic Tıp Laboratuvarı'nda Farmington, Connecticut, disiplinler arası bir araştırma ekibini yönetmektedir. Laboratuvar çalışmalarında gelişmiş genomik ve hesaplamalı modeller kullanarak bağışıklık sisteminin nasıl yaşlandığını ve yaşlanmanın aşılama ve tedaviye yanıt verme yeteneğini nasıl etkilediğini ele alan araştırmalar yapmaktadır.
Bioinfocongress II Professor Dr. Duygu Uçar Genomic Signatures of Human Immune System, Aging and Vaccine Responses. Professor Dr. Duygu Uçar got her bachelor's degree in computer science from Bilkent University, followed by her PhD in the same field at Ohio State University. During her PhD, she worked as a guest researcher at Technical University of Denmark. She conducted postdoctoral studies at the University of Iowa, founded by National Science Foundation NSF Informatics Innovation Fellowship, and at Stanford University Genetics Department. Since 2013, she is running an interdisciplinary research team at the Jackson Laboratory for Genomic Medicine, Farmington, Connecticut. Her studies how immune system aids and how aging affect our ability to respond to vaccination and therapy using advanced genomics and computational tools.
Dear Duygu Uçar and dear participants, I welcome all of you to Bioinfocongress II. Our esteemed guest will give us a speech on genomic signatures of human immune system, aging and vaccine responses. And at the end of the presentation, we will have a Q&A part prepared by our team and consisting of questions from our participants. Dear Professor Dr. Uçar, welcome again.
Thank you. It's a pleasure. Thank you for the invitation, Sema, and thank you for the nice introduction. I'll try sharing my screen. Let's see.
Right.
I think I did. Let's bring my slides. Okay, can you confirm that you see the slides?
Yes, now I can see your slides.
Okay, it looks good.
Yep, it does.
Okay, I can see myself, so I take your word for it. So I can get started whenever you are ready. Should I?
Yes, you can proceed with your presentation. Thank you.
All right. Well, good evening, everyone. I think most of you are in Turkey now, and thank you for choosing my talk over going out on a Saturday night. I really appreciate it. So I'll tell you a little bit about the research that's going on in my lab at Jackson Labs, which is in Farmington, Connecticut, halfway from Boston to New York, so we are close to both hubs. So today I'll tell you a little about our work in genomic signatures of human immune aging and vaccine responses. So I think Sema is going to help with me with the questions at the end. So I look forward to that as well. So I think this is no surprise to any of us, but aging is one of the major risk factors for complex diseases. So these are statistics from the US, from CDC, before COVID pandemic, but showing us the top 4 causes of death in the US. So as you can see, all the complex diseases here, heart disease, cancer, chronic respiratory diseases, as well as strokes. So what is common about these diseases, as we age, these diseases become much more dangerous for us. So I hope you are seeing my cursor, but as we get older, especially after 60 years old, the effect of these diseases on our lifespan is very significant. But also interestingly, this effect is not necessarily linear. So this line is not linear, and there are some time points in our lifespan where these diseases become much more lethal. And of course, in the last decade, I would say, the goal of the aging research has been shifting from not only increasing the lifespan of individuals, but also increasing their health span, meaning that we don't want individuals to live a very long life if they are going to be very sick. We want them to both live long as well as healthy as long as possible. So that's the distinction between lifespan and health span. So if the goal is to increase the health span, the immune system becomes a really important contributor, because almost all diseases of aging have an immune contributor. And let's briefly walk through what happens to the immune system and what are some of the basics of the immune system. So innate immune system, I don't know how many of you are familiar with the immune system, but very briefly, I'll tell you some basics. So immune system have 2 arms. One is the innate immunity, which is the fast and the first line of defense. So the innate immune cells are fighters, but they don't fight specific antigens. So they can fight all sorts of invaders, all sorts of molecules, all sorts of antigens that are not supposed to be in your body. And the examples of innate immune cells include monocytes, NK cells, dendritic cells. And the other arm of the immune system is the adaptive immunity, which is also known as acquired immunity. So this arm develops slowly, but once it develops, it has a memory, meaning that if you have already encountered a virus or an antigen, your adaptive immune cells can keep track of these. And then if you re-encounter the same virus, let's say, you develop faster responses and stronger responses. So for example, B and T cells, you're probably hearing a lot from Derya Unutmaz on TV, who is also in our institute. So some of these might be familiar to you by now, but B cells are the cells that make the antibodies, and T cells have different roles. Some T cells activate different immune cells. Some T cells are fighter T cells. So there are also different types of adaptive immune cells. So what happens to innate and adaptive immunity as we age? So we see a chronic activation of the innate immunity as we age, associated also with increased inflammation. You might have heard about inflammation in the context of diseases, but also in the context of aging. We see a systemic increase in inflammation. Some people call this inflammaging. And on the other hand, we see a chronic inactivation of the adaptive immunity. So the body is usually like a yin and yang, right? So if something goes up, something goes down. And in this case, the adaptive immunity is the one that is chronically inactivated. And this inactivation of the adaptive immunity associated with loss of naive T cells, increases in dysfunctional terminal differentiated adaptive cells. So together, these changes decreases our body's ability to recognize novel pathogens. And between these 2 changes, our immune systems become less and less protective as we age. In addition to not being protected from diseases, we also stop responding to available therapies or vaccines, which are much more effective in younger individuals. So after this brief intro, let me tell you a little bit about my lab's goals. So my lab is trying to uncover the genomic rules that govern age-related changes in the immune system. And a step forward, we want to understand how individuals are aging and why some individuals might be stop responding to therapy and vaccination. So the clinical outcomes are genomic biomarkers for age-related immune dysfunction, which might also give us ideas about how we can target some of these, let's say, dysfunctional pathways, dysfunctional cells for interventions to, in a way, rejuvenate the immune cell functions for older adults. And the strategy we are using is a systems level approach, meaning that we are not only looking at a single aspect of the cell, we are taking advantage of next generation sequencing as well as computational technologies to look at epigenomes, transcriptomes, proteomes, as many things as we can, and try to connect the dots by profiling immune cells from young and old volunteers. And we do this both in baseline, meaning that we just take samples from young and old individuals, compare them, and try to understand the effects of aging. But we also have vaccine studies where we study in longitudinal manner how the immune cells are responding to vaccination. So after this brief intro, let me tell you a little about some of the projects that have been published and some unpublished work in my lab. So let's start with this first question. What are the molecular biomarkers of human immune system aging? Of course, this is a big open question, and we have contributed to this work. We have been contributing to this work since I started my lab at Jackson Labs 8-9 years ago now. So this is our first paper that was published by my collaborator, Jacques Banchero, who's an immunologist, as well as George Kushel, who is a geriatrician, and he's leading the aging center at UConn Health Center, which is next to us. So how do these studies, I mean, broadly, these studies are also considered as systems immunology, because as I told you, we are taking advantage of next generation sequencing as well as computational biology to be able to connect the dots and to be able to cumulatively understand what's happening to the immune system, right? So in studies like this, you have multiple things that need to come together and that need to happen. And this all starts with a clinical enterprise. So we collect the blood samples from University of Connecticut Aging Center, which is next door to our institute. So in this case, we collected blood from 77 healthy individuals, 51 young and 26 older adults. So these individuals are screened for any confounding diseases, infections, medications, and frailty, which is a clinical score used to assess the health of older adults. So after, from the clinical enterprise, we access to these samples. In this case, these are blood samples. And then the first thing that we look at is the hardware of the system, which is the cell numbers by generating flow cytometry data. So in the blood samples, the blood-derived immune cells, also known as peripheral blood mononuclear cells, PBMCs, is composed of different types of immune cells. So I already told you adaptive and innate cells. So these cells, PBMCs, include both adaptive as well as innate cells. For example, T cell subsets, B cell subsets, monocytes, NK cells, etc. So the first thing that we look at is the hardware of the system. How are the cell numbers changing with age? And there are significant changes there. And on top of that, we also look at the software of the system. How are the cells functioning? And here the genomics become an important piece of the puzzle, because we can look at how the epigenomes are changing, we can look at how the transcriptome is changing, and then try to understand not only at the cell compositional level, but in the cell intrinsic level, what are some of the age-related changes. And of course, with a lot of big omics projects, the computational analysis is really important. And this is the Bioinfocongress, so I assume most of you are on board with this statement. So my lab is mostly computational, so my lab members have been working closely with the clinicians as well as immunologists to integrate and interpret all of these data. So I assume most of you are familiar with epigenetics, but let me briefly tell you what it is, just in case you are not familiar. So very briefly, epigenetics is telling us what parts of the DNA is open for business. So meaning that, so if you look at your DNA, of course, our DNA, other than the single nucleotide polymorphisms and mutations, other types of mutations, our DNA is very conserved across cell types, right? So if you look at the DNA from your brain cell versus DNA from your immune cell, they are pretty similar. But on the other hand, if you think about the functionality and even the phenotype of these cells, you know that they are very different. So this difference comes from the epigenetic layer, which means beyond genetics. So the epigenetic layer is much more dynamic compared to the genetics. So what is epigenetics? So your DNA, which is your genetic code, is very tightly packed inside the cell nucleus. So this tight packing is a research on its own, but one of the building blocks of this are the histone proteins. So here I try to represent the histone proteins. So the histone proteins are protein complexes around which the DNA is wrapping. And this wrapping keeps the DNA very compact, and only the regions of the DNA that will be activated can remove these histone structures, like this case. So here we have an inactive region of the genome, and in the active region of the genome, we drop some of these histone proteins so that transcription factors, which are proteins, can bond, and this can activate the genes, enhancers of the genes, and etc. So without this dynamics of the epigenetic layer, we won't be able to have all this variety in cellular functions, even the differences between individuals. So the epigenetic layer really acts like the software of the system and tells us which parts of the chromatin, which parts of the genome will be open, accessible, hence active. So the technology that we have been using since 2013 is the Ataxic technology. Very briefly, I already told you open versus closed chromatin, right? So very briefly, this technology is very effectively capturing which regions of the genome is open and accessible, hence functional. So the idea here is we have a hyperactive molecule called TN5 transposase, which is a molecule that can bind to the DNA if the DNA is open and accessible and cut the DNA. So what we do is we put the TN5 with cell type of interest and then identify the short sequences that are cut by the TN5 molecule. We align these to the genome, to the reference genome, which gives us at the base pair resolution, which gives us an idea about which regions of the genome is open and accessible, hence active, right? So here I'm showing you some example ataxic profiles, chromatin accessibility profiles. The top 5 are from healthy young donors, and the bottom 5 are from healthy old donors. And these are PBMCs, peripheral blood mononuclear cells, which include different types of immune cells. So this is a bit of a mixed bag. But with the, I know you had a training at the beginning of the Congress on the single cell data analysis. So with the current technologies now, we are able to do these kind of genomic profiling at single cell resolution, which really brings a lot of excitement and a lot of extra precision to how cells are changing with diseases, with aging, with vaccination, you name it. So it's really exciting to be able to analyze and generate that kind of data. Anyway, back to 2013. So this was bulk profiling, meaning that we did look at many cells, not single cells. In this case, we are using 50,000 cells. And the basic idea here is to first uncover open chromatin regions, also known as peaks. So for example, here I'm showing you a peak at the gene promoter of the Il7r gene. We will come back to this gene. And also we can capture chromatin accessibility regions, open chromatin regions that are in the non-coding site. So not necessarily at the coding regions or at the promoters, but non-coding regions. So these are Very important both for overall gene regulation, so the way genes are activated and inactivated are through these regulatory elements, but also if you look at GWAS studies, a lot of the disease-causing SNPs are following to these non-coding regulatory elements known as enhancers. So they are also inherently linked to genetics of the disease. I'm not going to talk about genetics today, but something to keep in mind. All right, so after we uncover these open chromatin regions, also known as the peaks, the next step is to uncover the ones that are differential between the two conditions, right? In our case, it's old versus young. So there are a lot of algorithms out there. We use generalized linear models, which are very well established for this kind of analysis. But this gives us an idea about which regions are more accessible, in this case, more accessible in young and less accessible closing with age, both at the promoter and in the non-coding space. So we have applied this pipeline to our 77 individuals from our first cohort, which gave us thousands, more than 12,000 differential peaks that are statistically significantly different between young and old individuals. And if we just make a heat map out of these differentially accessible peaks, this is the picture we get. So here we have a bunch of ataxic peaks that are more active in young individuals and closing with age. And we also have a bunch of regions that are activated with age, more accessible in older adults and less accessible in younger adults. So, of course, these are thousands of regions, and we did do a lot of enrichment analysis, but one region drove our attention from the beginning, and this was the Il7r gene and the genomic loci around this gene. So one reason that we found this very interesting is, first of all, this was among the most differentially affected in terms of epigenetic changes between young and old. What we have observed both in ataxic data, which is in the top, and the RNA-seq data, as we age, we see a decline in chromatin accessibility associated with decline in gene expression programs for this exact same gene. Which most of the time you expect them to be concordant, but in reality, it's not as beautiful. Anyway, so other than the fact that this was one of the strongest signals, there are a lot of important roles of Il7r. So let me tell you a little bit about the Il7r molecule. So Il7r is a cell surface receptor. R is coming from receptor. This is really important for any immunology studies, because if you have a protein that is expressed on the cell surface, you can do a lot with that. You can sort the cells with respect to that. You can look at which cells are expressing this, how many cells are expressing it, etc., and etc. So this Il7r receptor is when the Il7r is bound by IL-7, which is the cytokine, it activates the JAK molecules. These are kinases which can phosphorylate each other, and they can also phosphorylate other very important transcription factors known as STATs. So phosphorylated STATs, phosphorylated STATs can move to the nucleus, and these can bind to a lot of target genes. And this is important because then starting with this receptor, we can see a lot of downstream effect associated with phosphorylated STATs moving to the nucleus. So Il7r is a cell surface protein, as I already told you. In human cells, it's expressed on both naive and memory T cells, and it regulates response to the IL-7 cytokine. It's also important for the differentiation and survival of naive T cells, which are the T cells that have yet to experience an antigen. So when T cells are activated, they are not naive anymore. So inactivated T cells, which have the ability to recognize novel antigens, are known as naive T cells. And as we age, naive T cells decline. And this, which is one of the challenges of immune system aging. So this molecule is really important for the survival of naive T cells. So that's already some bad news for the T cell immunity with age. So when we look at the pathway, as I told you, this receptor doesn't function on its own. So when we look at the IL-7 signaling pathway, here you can see the image, and look at different components of it and whether we have observed chromatin accessibility changes with age. We noticed that not only the receptor itself, but a lot of the downstream molecules in this pathway has been linked to chromatin closing in our data, meaning that there could be a functional disruption in the T cells' ability to respond to the IL-7 cytokine. So, as I already told you, this is expressed on the surface of T cells. So this gave us an opportunity to understand which T cells are losing this molecule. So here I'm showing you data, flow cytometry data, which shows percent of IL-7 are positive cells and their percentages as a function of age in our cohort. So both CD4 helper T cells and CD8 killer T cells express this molecule at higher levels. But as you can appreciate from this figure, only CD8 T cells lose this molecule with age. CD4 T cells are not really affected with age in this pathway. So to close the loop, we also did a functional assay where we have isolated CD4 and CD8 T cells from healthy young and healthy old adults, and we stimulate these cells using IL-7 cytokine. So here we can see that, and then we measured phosphorylated STAT levels, which is downstream in this pathway. So these data showed us that if we look at CD4 T cells of young and old individuals, they have very comparable ability to respond to this cytokine. But if you look at the CD8s, which is the red bar here, you can see that as we age, our ability to respond to the cytokine is declining. So with this first study, at the time, this was published a while ago now in 2017. At the time, this was the most comprehensive chromatin accessibility map generated to study blood-derived leukocyte in the context of human aging. By using epigenomics, transcriptomics, computational approaches, we uncovered signatures of aging in CD8 T cells starting from PBMCs. And we have shown that this genomic decline is associated with loss of IL-7 responsiveness, specifically in CD8 T cells. So what are some potential implications of this? So there are already IL-7 treatments that can be administered pharmacologically and that can induce T cell functions with little toxicity. So these treatments have been already applied in HIV patients and have been shown to increase both CD4 and CD8 T cell counts. So there is a possibility that some of these already existing treatments might also help with the T cell dysfunctions that we observe in this study. So that was our first study, and of course we are continuing these efforts, and one of the big questions that we are trying to address is, do we all age similarly? This is a big question. I might spend my whole career trying to answer this question, but there is a lower hanging fruit here, which is whether there are differences between men and women immune system aging. I think all of you can appreciate that both in terms of health spans, lifespans, men and women are very different. So it is very logical to think that our immune systems might be also aging differently. So to properly study this, we again went back to our clinical collaborator, George Kushal's lab, and recruited more individuals. So in our first study, not by design, but women typically volunteer more to these studies. We had more female volunteers. So we went back and recruited more men, and we specifically matched these men to our female cohort in terms of any important clinical factors, in terms of ethnicity, in terms of frailty, as well as BMI. So in this second study, now we have many more individuals, 172 to be specific. So one other difference, in addition to adding more individuals, we also included individuals who are in the middle-aged group, because we know that aging is not a linear phenotype. So we wanted to uncover whether there are certain ages in the middle range, which is from 40 to 65, which is a long span. Whether there are certain ages where genomic changes become much more obvious. So again, we isolated PBMCs, profiled epigenomes as well as transcriptomes, and used flow cytometry to study cell compositions, and have been doing systems immunology research. So let's start with the cell compositional changes. I didn't tell you much about that in the first part, but there are some well-known changes to the cell compositions as we age for blood-derived immune cells. So one change is decline of T cells, so that we have observed here as well. So if you look at young, middle-aged, older adults, and we split the cohort based on females and males, we observe that there is a decline both in CD4 and CD8 T cells. I'm not showing you here, but it's in the paper. This is mainly stemming from the loss of naive T cells. A little bit more significant in CD8 T cells, but happens to both CD4 and CD8. We also looked at CD14 monocytes, known as classical monocytes. We didn't see major cell compositional changes here. Interestingly, in the B cell compartment, we have observed changes specifically to the males. We don't know exactly what's happening to these B cells, but it's something we will hopefully in the future clarify a little bit. This has been observed, not reported, but when we reanalyze data from another bigger cohort, we observe a similar trend. The B cells are declining, specifically in older men after a certain age. So going back to our favorite region now, IL7R, and what's happening to this region. So if we look at the same region, now we are looking at chromatin accessibility patterns cumulatively across the cohort and the IL7R expression. If we look at the profiles of females and males and summarize here, we see the declines both in males and females. But one thing that was obvious was the level of decline in terms of fold changes was much bigger in males compared to females. So although the same molecule is losing accessibility, losing much more in men compared to women. And this was not specific to just IL7R. If we look at the differential peaks, age-related differential peaks in men versus women, we notice that there are just many more differential peaks in men with age compared to in women. So twice, almost twice, more than twice differential peaks we were able to capture in men. So this is again bulk studies. We are moving into single cell versions of this, but in order to be able to deconvolute this bulk signal, we took advantage of roadmap epigenomics profiles. I don't know if you guys are familiar, but this is a big consortia effort where different immune cell types, many different cell types, but including the immune cell types, have been profiled using ChIP-seq assays, which gives us an opportunity to determine which regions are various cell type specific regulatory elements. So we use this reference database to deconvolute opening and closing regions with age, which gave us this, in a way, summary signature of aging in men and women. So B cells, I already described, probably this discrepancy is coming from cell compositional changes. What was really striking, and T cells were declining similarly, but what was really striking was the activation of innate immunity, particularly CD14 monocytes. So both in men and women, we noticed that the regulatory elements very specific to monocytes have been activated with age, but we captured 10 times more such regions in men compared to women. So the other piece of information that we were able to study is chronological patterns, because we collected data from middle-aged individuals. I'll come back to the activation of innate immunity, but let me tell you a little bit about this piece. So in order to do this, we did put together a computational pipeline that is starting with the batch-adjusted Ataxic or RNA-seq data. And then we are using a time series analysis method called autoregressive integrated moving average ARIMA models, which gave us an opportunity to select peaks that have a significant chronological trend. Then we clustered these regions, and then we functionally annotated them. So after applying this pipeline, we identified roughly 13,000 peaks, both in females and males, that we can cluster into 3 groups based on the chromatin accessibility patterns. So the first cluster, both in females and males, were losing accessibility with age. And when we look at whether these are enriched in certain cell types using roadmap chromHMM annotations, we observe that these are coming from mostly T cells. And then the second cluster, both in men and women, were activated with age associated with monocytes, and the third cluster was also activated with some differences in patterns, was associated with other innate cells, NK cells. So this also gave us an opportunity to understand and study whether these changes, these chronological changes in the genomic landscape, are linear or not. So we have spent quite a bit of time staring at these, and if you look at these as long as we did, you might see that these changes are not necessarily happening very linearly over time. So you can see that some time points in these patterns become much more different compared to the previous ones. So we call these breakpoints, which are the age brackets associated with sudden genomic changes. And to capture these breakpoints, Eladio Marquez, who was in my lab at the time, established a simple pipeline. So here the idea is you select an age here, and then you compare the epigenome before and after this age, and we change the window. It could be 5 years before, 5 years after, 10 years, etc. But here the idea is to compare the epigenomes in this 5-year period and in this 5-year period and associate a p-value if they are very different. And then we just proceed, do it for the next time point, etc., etc. So if we span the whole age bracket in our cohort and do this p-value calculations, we get these interesting signatures. Of course, this has to be done at the cluster level. That's why there are 3 lines here. But what we have observed here is the fact that there is one break point early in late 30s, early 40s, both in men and women. And this is very comparable between sexes in terms of p-values. But the second breakpoint is much more different between sexes because it first happens earlier in men, roughly 5 years, and the p-values are much more significant, meaning that the genomic changes are much more abrupt. So what would this even mean? Yes, this is computationally interesting, but does it have a meaning in clinic? We don't know yet. But if I go back and remind you the CDC data, this is strikingly similar. Of course, in the CDC data, we didn't split the database on sex, which we should have done. But if you just look at these patterns, so roughly the time points here where the chronic diseases start killing us and accelerated effects of these diseases are matching our breakpoints. So the other piece here is comparing men and women, right? So if you go to a doctor, especially if you are there for a chronic disease, they usually don't take into consideration your sex when the doctors are prescribing therapies. But it is probably something that's going to change in the future because our genomes, epigenomes are very different, and there is a lot to learn here. So to understand whether there are molecular differences between men and women at different ages, we compare the epigenomes and transcriptomes of men and women, excluding sex chromosomes where we expect to see differences. So these are MA plots. Here the information, every dot is an ataxic peak, and the x-axis is the size of the peak, y-axis is the fold change. But what is important here is the ones that are colored with red or blue are the ones that are statistically significant between men and women. So what we have observed here was a little bit different than what we expected to see, because we were thinking as individuals are younger or as they are going through big hormonal changes, menopause, etc., there will be more differences. But in contrast to what we expected, we see more differences in older adults, so after 65 years old. So that was kind of interesting. And we observed this both in epigenomes and then the transcriptomes, we see the same thing. So what are these changes? So when we annotate them, so again, this is bulk data. In order to deconvolute this, we identified gene sets from single-cell RNA-seq data, which are very cell type specific, and then looked at female and male bias regions, both in Ataxic and RNA-seq, and associate them to a cell type. So this told us that both in epigenomes and transcriptomes, older men have more active innate immunity, particularly in monocytes, and older women have more active adaptive immunity, B cells and T cells. And if we look at some of the pathways associated to these sex differences, we again observe some of our favorites. So for example, women had more active IL-7 signaling pathway compared to men, whereas men have more active inflammatory pathways compared to women. And from our differential analysis, we know in which direction these are going with age. For example, IL-7 signaling is declining both in men and women, whereas inflammation is increasing in both men and women, which is suggesting that in some of these pathways, although both men and women are experiencing similar changes, the rate of activation or the rate of inactivation could be different. So, of course, the story of monocytes, further activation of monocytes in men are interesting, and this could be linked to inflammation. So one way to measure inflammation is from the serum, you can look at pro-inflammatory cytokines, which we have not done for our cohort, but we have found data from another study, 500 functional genomics, and we compare the pro-inflammatory cytokines of older men and older women from this independent cohort. And as our genomics data suggested, we have noticed that in this separate cohort as well, older men have more pro-inflammatory signatures compared to older women. To summarize this part, so this is a story that we published just before COVID happened, but here we observe the fact that inflammation is increasing both men and women, but these increases are much, much stronger in men. And on the other hand, the adaptive cell declines are much more significant in men compared to women. Also, by doing some time series analysis, we show there could be also some differences in the timing of changes. So men are going through 5 years earlier to these changes in our cohort compared to women. And clinically, probably the most interesting part is male and female blood-derived immune cells diverge after 65 years old. Older women have more active adaptive immunity, whereas older men have more active innate immunity. And the implications of these for health and lifespan, of course, there is a lot to learn here, but let me remind you some already known sex differences. So in the US population, so for example, men are more susceptible to infectious diseases, and typically they experience more severe infectious diseases. For example, COVID-19 is a good example where you see 1.5 to 2-fold higher death rates for men compared to women, depending on the country. On the other hand, women are more susceptible to autoimmune diseases. Lupus is a good example where 90% of patients are women. Another aspect of sex differences is in the immune responses. So how do men and women respond to different immune challenges, including vaccination, is a little bit unknown, but typically women better respond to influenza vaccination, yet experience more adverse reactions. However, there is a lot to learn in this immune responses and sex differences in immune responses. And that's an area that we are actively working on. And in the remaining of my time, I'll tell you a little bit about this unpublished study. So our study uncovered that women have higher adaptive immunity and lower inflammation, but does it mean that they can respond better to vaccines? And long-term goal, of course, can we predict who will respond to which vaccine? So you might have heard about the precision medicine terminology, where instead of giving individuals random therapies, we try to give them very precise, personalized medicines. So this is a little bit like that. We can call it precision vaccinology. So we give the individuals the right vaccine, right? So this became much more interesting after we have many different types of COVID vaccine, but it was interesting even before that. All right, so this is one study that is still ongoing, but this is a vaccination cohort for bacterial pneumonia. Of course, with COVID, we are thinking more about viral pneumonia, but bacterial pneumonia is still a big health issue, especially for older adults. In the US only, thousands of individuals are hospitalized or die from this disease. And for older adults, the rate of death is pretty high if you get the invasive disease. There are 2 FDA-approved vaccines for this in the US. So one is Pneumovax, which is a B-cell-mediated vaccine. It has a polysaccharide. And the other one is the Prevnar. So Prevnar is a vaccine that has the polysaccharide that induces B-cell responses, but it also has an adjuvant protein that induces T-cell responses. So the current guideline in the US is to give individuals Pneumovax after 65 years old, and Prevnar is only used for vulnerable populations like individuals who are living in nursing homes, etc. Okay, so very briefly, I don't have much time left, but very briefly, the 2 vaccines, the differences between the vaccines, I already told you. There is a polysaccharide that induces B-cell responses in the case of Pneumovax. In the Prevnar, you have the polysaccharide and you have a protein associated with it or connected with it. So this protein induces also the T-cell responses. The other difference is the number of serotypes, number of different bacteria strains. So Pneumovax have 23, Prevnar has 13 of these major differences. So the way we do the, I forgot to tell you, but when we do the vaccination studies, these time points become really important. We look at the baseline because baseline gives us information about the status of the immune system before individuals even got the vaccine. If there is a baseline predictor, this is really important. Day 1 is usually important for innate immune responses. Day 7 to 10 is important for adaptive responses. Usually 1 month after the vaccination, we look at the antibody titers, or in this case, OPA titers, to understand whether your ability to kill a bacteria or a virus is increasing upon vaccination. And day 60 is important to understand the memory of the immune system. All right, moving to how we quantify vaccine responses, right? So this is an essay from our collaborator Moon Nam's lab. It's a very functional essay, in fact, which is also known as opsonophagocytosis assays. So here the idea is you get the vaccine from the individual as well as the bacteria, and you put these together and see what percent of the, or how much serum do you need to kill 50% of the bacteria. So you just change the dilution of the serum to understand at what point at least 50% of the bacteria is killed by the phagocytes. And we do this before and after vaccination to understand how much we are improving your serum's ability to kill the bacteria. So this is a little bit of more complex than your regular influenza vaccine, which only have 2 or 3 strains. Here we have 13 strains, right? So every column here is a strain, and every line here is an individual. So for every strain, for every individual, we are quantifying their blood serum's ability to kill bacteria before and after vaccination. So if there is a threefold change in log2, we call this responsive. But as you can imagine, this is a complex data, and we came up with some computational ways to also quantify these responses. So we look at the strength of the responses, the sum of responses to all serotypes. If you have higher values, that's better. So you are developing much stronger responses. We look at the extent of responses, meaning that we are looking at how many serotypes an individual is able to develop strong responses. And using these 2 measures, we also rank the individuals in terms of their vaccine responsiveness. So the individuals that have higher ranks are the most responsive. So here, for example, this first line is the most responsive in our cohort. What's really striking in older adult cohorts is there is just so much heterogeneity in the individuals. So there are some individuals, although they are all older than 60 years old, some individuals who develop really strong responses and some individuals who do not. So I told you briefly that one way we want to use this data is to understand who can respond to which vaccine. So we have collected flow cytometry data, which tells us what type of immune cells we have in the blood of individuals. And by using this data, we correlated responsiveness to vaccine to different cell types. And one particular cell type, in fact, 2 cell types, and their ratio was the most predictive for Prevnar vaccine responses. And this is Th1 to Th17 ratio. Th17s are pro-inflammatory T cells that produce also very inflammatory IL-7 cytokines. Th1 induces antibody and cell-mediated responses. So if you have more Th1 and less Th17, you develop stronger responses to Prevnar. But for Pneumovax, none of the cell compositions, and this is just to quantify that, none of the cell compositions were predictive for Pneumovax. For Prevnar, PCV13, these 2 were very predictive. So briefly, when the T cells, CD4 T cells, are activated by an antigen-presenting cell like a dendritic cell, they can develop into different cell types. And this is mainly determined by the existence of other molecules in the cellular environment. So the way Th17s differentiate, the way naive T cells differentiate into Th17 happens in the existence of pro-inflammatory cytokines, IL-6, IL-1B, etc. Meaning that if you already have a very pro-inflammatory immune system, you might have more Th17 cells. In fact, there have been studies showing that with age, there is an increase in Th17 levels. So I already told in the second part of the talk, men have higher inflammation, right? So then we looked at whether this has anything to do with their sex vaccine response differences. So if we looked at responsiveness, again, remember that higher is better responsiveness of individuals to different vaccines. We noticed that women develop stronger responses to Prevnar, whereas there are no sex differences for Pneumovax. And does it have anything to do with the Th1, Th17 ratio? In fact, it does. So if we look at Th1, Th17 ratio of men and women in our cohort, we observe that typically women have much, much higher levels. Interestingly, this didn't correlate, Th1, Th17 ratio didn't correlate with age in our cohort, and maybe it's because these individuals are already older than 60 years old. So to summarize and to tell you a little bit about where do we go from here, so we observe in our first study a dysfunction in IL-7 signaling cascade. We are very interested in understanding, can we rejuvenate immunity in older adults via treatment and intervention. We have an ongoing study where we are studying the single-cell transcriptomes from older adults who have gone through a very significant lifestyle change. We observe men and women immune systems age very differently, and in relation to what we are observing, we show that older women respond better to T-dependent Prevnar vaccine. And our study also uncovered older men have higher innate immunity, and this tells us we should be considering sex while administering therapies and vaccines to individuals. And also in the last part of the talk, I show you we can predict responsiveness to Prevnar vaccine with a simple test. And as I already explained, this is an opportunity to precisely administer vaccines to individuals. So that is my last slide. So complex problems require multidisciplinary solutions, and my team is also very multidisciplinary. So the work I presented to you are done by Eladio, Onur, Fernando, Cheng Hang, and we are continuing some of these efforts. Satya is continuing the work in vaccine responses. And if you are interested, we are hiring computational and experimental scientists at all levels. Just send me an email, send me your CV, and we will just discuss. And both my collaborator, Jacques Banchero, who's an immunologist, as well as George Kushal, who's a geriatrician, have contributed significantly to the work I presented to you today. All right, I have a little bit of time for questions. Should I stop sharing or should I?
Of course.
All right.
Thank you very much for your presentation, Professor. If you wish, we can continue with the question and answer part.
Sounds good.
All right, and the first question is, can modulating T-cell differentiation be an effective approach to increase vaccine efficacy in elderly individuals?
That's an interesting idea. Potentially, yes, but we don't know the implications of this for other diseases or other conditions, right? Potentially, yes. It's probably something that one would start looking in mice before we move into human. But potentially, for example, I told you the molecules IL-6, IL-1B, which are very pro-inflammatory. So there are already some anti-inflammatory treatments that tries to reduce the level of these pro-inflammatory cytokines. So the effect of these anti-inflammatory drugs on vaccine responses is not studied yet. But it's an interesting question. So if we get an anti-inflammatory drug for a month before we get vaccinated, do we develop stronger responses to certain vaccines? I think that's a very interesting research question. If you join my lab, we can write a grant on that.
Thank you for your answer. And the next question is,
Gamma delta T cells, I see the question. Well, in our study, we didn't, gamma delta T cells are also not as frequent in the blood as some others, but now with single cell technologies, we are able to see them. So in the bulk data, we cannot really separate all these subsets of the data. We can do the flow, but honestly, it hasn't been our top candidate. You are right, it's a little bit neglected. But with single cell technologies now, both in human and mice, in tissues, as well as in blood, we see a lot of gamma delta T cells. And some of them are really associated with pro-inflammatory conditions. We see them in COVID as well. So I think there will be more to learn there, and single cell technologies will help.
Thank you for your answer. And the next question is, how does genetic tendency protect the immune system against aging?
Genetic tendency. I interpret this question as whether our genetics can protect us from immune system aging, or whether some protective variations possible. So if you look at Dr. Casanova's work, he is a professor at Rockefeller University. So he has, for example, associated some, what does he call them, inborn errors, which are mainly variants in certain interferon response genes to, for example, COVID-19 severity. So it's possible that some of us have some really protective genetic variants that protects us from pro-inflammatory diseases like COVID-19, but can also help us in the context of immune system aging. So it's possible. We have an ongoing study where we will look at influenza vaccine responses, and we will also genotype the individuals. It's still a relatively small cohort, but it's possible that we might see some associations.
Thank you for your answer. And the next question is, how aging-related T-cell dysfunction can affect cytokine production while impairing immune tolerance mechanisms?
Oh, that's a loaded question. How aging-related T-cell dysfunction can affect cytokine production while impairing immune tolerance mechanisms. So I don't know exactly what mechanisms you are referring to, but so for example, some of the T cells, this is much more studied in mice aging. for example, there is this population of granzyme K positive T cells described by Max Artyomov and his team, and we see it in some of our studies as well. So these are, in a way, dysfunctional CD8 T cells, but they produce a lot of interferon gamma, and which is potentially activating the monocytes and innate immunity, right? So different types of immune cells, even if they are dysfunctional cells, they can produce cytokines or secrete different proteins, which can activate different types of immune cells. So that's how the whole immune systems connect to each other, they talk to each other. So one change in one arm of the immune cells can definitely affect the rest of the immune system. I hope that helps answer your question.
Thank you for your answer. And the next question is, what is the impact of frequent high nosocomial infections on immune aging in older adults?
I don't know what nosocomial infections means, if you know either.
It means when you catch a disease, an infection from a hospital you stayed at.
I didn't get that question. I'll pass that one.
All right. Next question is, what are the goals expected to be achieved in the future with the use of bioinformatic analysis on the human genome for the development of the immune system?
The goals, I try to give you a flavor in my talk. So the goals is to understand precisely how the individual's immune systems are. And this is not necessarily in the context of aging, right? So there are a lot of immune diseases. There are other types of things. For example, obesity has been linked to increased inflammation. So other factors that might really make our immune systems weaker. So my goal personally is to use all these single cell technologies, advanced genomic computational methods to precisely understand how our immune systems are functioning and dysfunctioning in disease and with aging. And ultimately to be able to link these dysfunctions, pathways, molecules, etc., to available therapies, and to be able to more precisely treat individuals when they go to the clinic.
All right, thank you for your answer. And the next question is, what is known about the ability of vaccines to form tissue-resident memories with age?
Oh, that's an interesting one. I don't know much about that, to be honest with you. Yeah, we are not really looking at this point, tissue-resident cells, because it's more tricky to get these in human, right? And also, which tissues are you gonna start with? Probably the immune organs like spleen, lymph nodes, etc. But again, it's very invasive in human. Probably mouse is a good starting point to answer that. But there might be other studies published from other labs, so you can probably find answers elsewhere.
Thank you. And the next one is, what is the hierarchy of transcription factor binding to IL-7 regulatory elements in different types of cells?
So I am not sure what you mean by hierarchy of transcription factor binding, but the STAT transcription factors also bind to the IL-7R promoter. So it's a little bit like this loop that IL-7R activation can activate STATs, but STATs then go and bind to the promoter of the IL-7R. So it's a little bit this circular loop. So the transcription factor binding, just to clarify, doesn't change that much from cell type to cell type because it's the genome, right? It's the sequence. So transcription factors bind to certain sequences at the promoter of the genes, let's say, and that sequence is not changing. What changes is whether that transcription factor is expressed in that cell type and whether that region is open and accessible. So if the region is open and accessible, if that transcription factor is expressed in one cell type, it's going to bind similarly, even if it is immune cell or brain cell, and etc. In the case of IL-7R regulators, STAT1 is one of them. There are a few others. There was a figure in our paper. If you are interested, you can go and check that out. But yeah, I don't know exactly what you mean by hierarchy, but the order of binding, we don't really know if that's what you mean. If one transcription factor binding first, if the other next, we don't know much about that binding, but it could be relevant as well.
Thank you. Can we identify the cells that are stimulated to secrete IL-7 during immune responses against certain diseases?
Oh, that's a good question. Yes, we can. I should know the answer to this. I think monocytes are mostly secreting IL-7. I'll check that out. Yeah, this is, I mean, it's never a one-to-one equation, right? Some cytokines are secreted by multiple cell types, but in the case of IL-7, it could be monocytes. I'll check that out. But we can do that. Yes, we can do that. That's very doable. In fact, there are single-cell proteomics assays now that can bring more resolution.
Thank you. Is assessing chromatin accessibility in human immune cells advantageous over other genome-wide measures?
So the epigenome of the system tells you the potential of the system, because if you look at transcriptome, the gene expression level, you only see the things that are active at a given time for a set of transcription, for a set of region, right? So epigenome is in a way the potential of the cell, what could be activated, because there are epigenetic signatures that are primed, meaning that the epigenetic layer is open and accessible, but the transcription has not happened yet. So there is that benefit of epigenetic profiling. Among the epigenetic assays, honestly, Ataxic is my favorite. DNA methylation, there are a lot of DNA methylation clocks and etc., but it has been very difficult to link those CPG sites to nearby genes. What I like about Ataxic, as I showed you in the IL-7R example, you can really uncover a regulatory element which is very close to a gene and obviously regulating the activity of that gene, right? So it's very easy to identify an epigenetic signature from chromatin accessibility and link it to a gene and then study that gene and the pathway and etc. associated with that gene. So for those regions, that's one of my favorites.
Thank you. And the next question from YouTube is, now I can see it, all right. What is the main reason why men's immune response differs from women's? Is there a difference in immune response between women who have given birth and those who have not?
Good questions. I could probably write grants on both. So the first question, we don't know yet. It could be multiple things. One idea that I'm exploring now is whether the trained immunity could be different. So trained immunity meaning that if your immune system is encountering some previous challenges, this could be vaccination, this could be disease, and etc., there is an epigenetic memory left in the stem cells in your blood or stem cells in your body. So this is important because monocytes, for example, do not have a long life, so they die quickly. So if there is a chronic ongoing inflammation all the time, so there are like serums, cytokine levels are very high for pro-inflammatory cytokines, or the newly generated innate cells have a more pro-inflammatory signature. And this could happen through the stem cell population. If there is an epigenetic memory of previous infections, previous responses, stored in the stem cell population, and if this is different between men and women, then they might have very different pro-inflammatory profiles, for example. So that's potentially one difference, whether the HSCs, stem cells, are aging differently and whether they could be leading to these differences in inflammatory signatures. So I'm exploring that with a collaborator, or at least planning to. I'll report back in the future. So given birth, that's a good question. So in our cohort, we didn't really collect that information. it is a separate study where one needs to really recruit individuals, women who have given birth, who has not. There are a lot of parameters there, right? So does it matter how long ago one gave birth? Does it matter if the individual were in 20s, 30s, 40s when they give birth? So there are a lot of things that one needs to carefully design a study there, but it's possible. I mean, giving birth is potentially a big thing for human body and potentially affecting the immune system. All right.
Thank you. And the next question. How does the changing epigenetic profile with aging affect the response of older individuals to vaccines?
We don't know. I wish we do. You can change epigenetics in different ways. So there are versions of CRISPR, CRISPR-I, CRISPR-A that do not change the genetic sequence, that can change the epigenetic layer on top. So it would be interesting to study those in the context of vaccine responses. One thing that we do know, though, from Bioinfoconference studies from Stanford, so what he has shown is that so there is an epigenetic memory associated with vaccine responses, let's say to influenza vaccine response. And he has shown that if the individuals get a second vaccine, when this epigenetic memory is still in place, they develop stronger responses. So this is kind of suggesting that if the epigenetic memory associated with a vaccine response is still intact, it can potentially boost the immune responses to an even irrelevant vaccine based on his studies. It's a very interesting paper. I recommend that you read it from Bioinfoconference.
Thank you. And the next question we have is, how do cestrins affect the function of CD8 plus T cells?
Good question. I don't know. We have not really studied that. It's an interesting question. Probably requires a cardiologist's help.
All right, and that was the last question. On behalf of myself and our participants, I would like to thank you for spending your precious time with us.
It's my pleasure. I thank you for spending your Saturday evening with me. And I know Emek is the next, and I know him from Bilkent and from US, so you are in good hands. You are up for another treat. All right, bye.
It's great to hear.
Have a good one.
Bye. I wish you a healthy day. Thank you again.
Bye-bye.